Hello students, so now we'll learn what is spermatogenesis. So spermatogenesis is all defined as a process of development and production of sperms in the male reproductive system. So like oogenesis, it is also a very important process because if there would be no sperm, there would be no fertilization of ovum that was produced in the female reproductive system, and if there would be no fertilization, there would be no beginning of a new fetal life and continuation of the progeny. So let's see what are the stages of spermatogenesis. So spermatogenesis is divided into three stages: cell division, spermiogenesis, and spermiation. So cell division is the first stage of spermatogenesis, where the stem cells get divided mitotically and meiotically and finally produce four spermatids. And spermiogenesis is the second stage of spermatogenesis, where the spermatids produced undergo development and differentiation and finally become like a typical sperm. The third stage of spermatogenesis is spermiation, where the developed sperms migrate to the lumen of the seminiferous tubules for storage. The entire process of spermatogenesis takes place for about 65 to 75 days, and it starts in the seminiferous tubules at the basement membrane, and it starts from the stem cells called spermatogonial cells. So let us see the first stage, that is cell division. So initially the spermatogonial cells, which are the stem cells, undergo mitotic division and produce many spermatogonial cells. Some of these produced spermatogonial cells get attached to the basement membrane of the tubules and do not divide further. Instead, they get stored there for production of sperm in the later stages of life. But some spermatogonia undergo differentiation to develop into a primary spermatocyte. The primary spermatocyte that is developed now enters meiosis 1. The primary spermatocyte is a diploid cell consisting of 46 chromosomes. So here the two pairs of, in meiosis 1, the two pairs of homologous chromosomes get attached or get aligned on the metaphasic plate in a single alignment or in the same alignment. So here in the metaphase 1, when they align on the metaphasic plate, crossing over takes place. What is crossing over? So this is exchange of certain segments of genes on the chromosome with the segments of genes on the other chromosome. This is the reason why we are a little different from our parents. So once this is completed, in the anaphase 1, the one chromosome of each pair, or one chromosome is pulled to one side and the other chromosome of that pair is pulled to the opposite side, and finally the two secondary spermatocytes are produced, which are now haploid. That is, they consist of 23 chromosomes. Then the secondary spermatocytes which are produced enter meiosis 2, where they again meiotically divide. Now they consist of single chromosome. So they do not exist in pair because they are haploid. So now each chromosome again in the metaphase 2 gets aligned on the metaphasic plate and gets aligned on the metaphasic plate, and these two chromatids on the chromosome are attached together by a centromere. So in the anaphase 2, the chromatids are pulled on the two chromatids are separated from the centromere and they are pulled to the opposite side. So like this, the meiosis 2 is also completed, and finally in this meiosis 2, four haploid spermatids are produced. So this is cell division. But one major difference in spermatogenesis and in the normal meiosis is that here cytokinesis is not completed. Do you remember what is cytokinesis? It is the final stage of any cell division, where the daughter cells which are produced are completely separated from each other by cleavage in the cleavage furrow that is formed in the telophase. But here this cytokinesis is not completed, and therefore the four spermatids that are produced are attached together by cytoplasmic bridges. Next, the second stage of the spermatogenesis is spermiogenesis, where the four spermatids that are produced from one primary spermatocyte undergo differentiation and development and become like a typical sperm. Now, what is this differentiation or development? The rounded spherical spermatids become elongated and slender. Then the acrosome is formed at the head of the sperm or the cell, and the flagellum is attached to the tail of the cell. Excess of cytoplasm is removed, and the number of mitochondria increases. Now, number of mitochondria increases because a lot of ATP or energy is required for locomotion of the sperm in the female reproductive system. The flagellum also serves for the same function, that is locomotion. It is the only cell in the body that can move or that requires locomotion, and that's why it is the only cell that consists of a long flagellum. The last step of spermatogenesis is spermiation, where the developed sperms are broken off from the cytoplasmic bridges. They get separated and ultimately migrate to the lumen of the seminiferous tubules of the testis and get stored there. So quickly we'll revise what was spermatogenesis. So initially the stem cell was called spermatogonial cell, which got mitotically divided and produced many spermatogonial cells. Then some of those spermatogonial cells were stored and some others were developed into primary spermatocyte. Now the primary spermatocyte entered meiosis 1 and produced 2 haploid secondary spermatocytes. The secondary spermatocytes entered meiosis 2 and produced 4 haploid spermatids. This was cell division, and then the 4 spermatids that were produced underwent development and differentiation and became a typical slender elongated sperm with an acrosome at the top and flagellum at the tail with many number of mitochondria within them, so that much amount of ATP can be produced. So this step was called as spermiogenesis. Ultimately, once the spherical spermatids were developed into complete sperms, these sperms got detached from their cytoplasmic bridges and became free individually, and then they migrated towards the lumen of the seminiferous tubules and got stored there. Any doubt, you can again clarify anytime. Read notes carefully and listen to this carefully. Thank you.